help us to start mapping and tagging all the genes per dwarfing genes which help the green revolution, rice and wheat. And we found similar kinds of gene in pigeon pea also. And now we are developing these varieties which are under the testing. And then we made a chip in pigeon pea also. We made a chip for 62,000 SNPs. And this chip has been very useful for doing the mapping, all those things applications. I won't go into the detail. Jute is another crop. India and Bangladesh. It's a very large volume cash crop for India and Bangladesh. Nobody else will put their money in that. So we took this one, and then we have again published the jute genome, and we mapped the gene for the fiber quality. So these are the two papers here. Then let's come to the last one, which is the mango. Mango is actually known as the king of fruit in India here and many other countries also, because it is a very large volume, $10 billion value commodity this one is. But we hardly export anything because we don't have much breeding work, not much work on the quality. But if somebody will eat the Indian mango, they will not forget the taste. They will keep on like to eating it. But how to make that commodity export worth exporting, we need to put a lot of science in that. Again, India is the topmost producer and consumer. Mango has originated in India. There is no controversy about this, because we have 70 million years old fossil record in Meghalaya, and then mango has gone from India to rest of the subtropical world under recorded history. You don't need science for that, it is history. That on which date it went to Southeast Asia, to China, Australia, Africa and America. So there is no such controversy. We have more than 1,000 varieties of mango of all size, color, taste and thing, and the detailed characterization has been done by the horticulturist, but there was no genomic tools and these things available. So we took up this challenge of sequencing the mango variety Amrapali, which is the first hybrid variety, which is in the orchards, mango orchards. But when we assembled this genome using the Illumina reads, which are very short reads, we find the genome assembly was much larger than the estimated genome size. And then we actually did the genome size estimation again by flow cytometry, and we see that there was nothing wrong with the genome size estimation. Then how the assembly is coming larger than the genome? And instead of that, the answer to that was that it has very high heterozygosity. So maternal and paternal genomes were assembling separately. Wherever the heterozygosity was more, you are having those countries, they don't merge. And that's why then you need to find another solution. And then the third generation sequencing came to our rescue, which is the PacBio sequencing, which is a single molecule sequencing. You don't need to do even PCR, you just need only the DNA and sequences. Now the Oxford nanopore is also coming. So here, because of the very long reads, and we generated the additional actually reads for that. So time is short, I am just summarizing. We now have got this assembly which is 403 MB long, which is almost 95% coverage of the genome. And it was possible to assemble this heterozygote genome because we can now go for very large overlap because of the longer reads and allow too many mismatches, which takes care of the heterozygosity, which is not possible with the short reads of Illumina. So as a result, we have submitted this sequence, and the genome coverage is very high. This genome was anchored using a map published by China. They published a very high density map of more than 6,000 markers in mango, and this was used to anchor the genome. All the 20 chromosomes have been anchored like this, and we have predicted 46,000 genes, and we find that the highest similarity is with Citrus sinensis. So the sweet orange has got the highest similarity with the mango. One is sweet, and then we can draw up the phylogenetic trees and all those things, routine things. We have annotated the repeat elements in that. You have DNA transposon, simple repeats, low complexity, the non-LTR. Then we have annotated telomere, centromere, and non-coding RNA. Then we have found segmental duplications in that. Like many other genomes, the mango has also gone through the segmental duplication, it's an ancient polyploid, and you can date all those duplications when they took place and what relevance they have. The heterozygosity was worked out is very high, and every 38 base pair there is an indel or SNP. That was creating problem for the assembly with the short reads. Now again, We have created a chip for mango also, now this time 82,000 SNP. So all these resources, tools are going to be very useful. This one example here, the pulp percentage. Like the seed will be small and there will be more pulp. We found 3 major QTLs here, which is mapped by association mapping with very high log value. So second last slide is that. Now what are the future challenges and opportunities? Now we have the genomics technology coming up very fast, is getting cheaper, and more development is taking place. We also have the optical mapping based on the single molecule optical mapping. So we can use these technologies to create the reference maps of the species which have got very high significance for India or for any other country who have who can put their money. Then we combine these things, single molecule technology now. We don't need to go back to these older technologies. Then also iso-seq, their technology, you can have full length RNA transcript or splice variant. All those things can be combined with the high genetic map. So I just have given you these 3 examples, but what I'm trying to say, that similar thing we can do with the other species also. We have many plants, fruit, horticulture and medicinal plant. This thing can be done, and then one can make use of that in developing better variety or preserving our germplasm or through IPR, and then identify new genes and markers, which is the ultimate purpose, that all this science should serve the society. Whatever science, great science we are doing. So with the vision of Dr. Khurana, that unless it is serving the society, it is of no use. So we hope that I have given you the glimpse of what we are doing, and in what is the future, what more things we can do, maybe young people will do. There are so many things to be done, and the time is the limiting factor. So I will stop here. Thank you very much for your attention.
Thank you very much, Dr. Singh. Do you have time for any questions?
Yes, if there are any questions, I'm happy to. No question?
Any questions from the audience? Okay.
I can ask that. Many of the QTLs you have already identified in rice also. Some are responsible for salt tolerance, some are for yield also. So the genes which are actually there in the markers has also been identified?
In some cases, yeah.
But
in most of the cases, genes are not identified.
So that work is still pending.
Yeah, but we can still use it in the breeding, because if it has narrowed down to very small segment of maybe 200 kb or 300 kb, we are using them in the breeding. For drought tolerance, we have almost half a dozen QTLs. We have transferred in these high yielding varieties, and they are now drought tolerant. Submergence tolerance gene is known. Salt tolerance, some genes are known. So this is the stepwise we have to proceed. Even before knowing the gene, we can make use of that.
Any other questions? If not, perhaps we'll carry on with our, thank you, Dr. Singh. Will you join me in thanking him again? We'll carry on with our program. Our next speaker is Dr. Jitendra Khurana, not Khurana, who obtained his PhD in botany from the University of Delhi in 1982, and he did postdoctoral work at the prestigious Smithsonian Institution in Washington and at Michigan State University. He returned to Delhi to join the newly established Department of Plant Molecular Biology at South Campus in 1988 and has been there ever since. He'll be telling us today about some interesting gene targets in plant development. And I think it's rice development.
Thank you very much. I must thank Dr. T.R. Sharma for giving me this opportunity to be a part of this very interesting symposium. I must also clarify, this is the same surname. Okay. It's not mine. Okay. So this is spelled differently. In fact, more than 99% Khurana is spelled U, not O. But then Hargobind Khurana was different in many ways. Well, as Dr. N.K. Singh said, that some of us really grew up listening to the contributions of Hargobind Khurana. And interestingly, during my undergraduate days also at Delhi University, during our exams when we were taking practicals, whether that was in anatomy, physiology, morphology, rather than asking questions in that area, everybody will ask me, the teacher, what is the contribution of Hargobind Khurana? So this all went through almost from 1970, 71 to 75 till I did my master's. And in fact, that's what also motivated me to take up science as a career, although in plant sciences, not in medical sciences or other areas where Dr. Khurana worked in. But this is interesting that when I moved in at Michigan State, and also before that at Smithsonian Institution, I moved on to work in an area where one was interested in finding out how plants perceive light to regulate their development. So in that sense, I think later part of Dr. Khurana's contributions or adoption, in that sense, I think relates very well in the area of photobiology and signal transduction, the area which I also work on, although in plant systems. Well, most of you in plant sciences background know that most of the contributions, understanding about plant development has come through work on Arabidopsis. And as Dr. N.K. Singh mentioned, now rice is a model system for monocots or cereal crops. And in my lab, the focus has been on whether these cereal crops are developing in a different way than dicots like Arabidopsis, and whether some of the genes may have acquired novel functions to regulate their development. So I'm going to just be very, very focused on trying to focus on one particular transcription factor, which is a basic leucine zipper. And this is the one which is, So this Hi5 is a basic leucine zipper transcription factor which was identified in Arabidopsis many years ago, and has turned out to be a very central regulator to downstream to many different kinds of receptors in plant systems. So the work which I'm going to be describing in today's presentation is done largely by Naini and Akanksha. Other people have done other work which I don't have time to talk about. Well, since some of you are from the area, you may not be familiar that how exactly plants perceive light. So I have just 3-4 slides to make a general introduction. In the last 50 years' time, it has become known that there are 3 major classes of sensory photoreceptors: phytochromes, which scan the red and far red region of the solar spectrum, and blue and UVA part of the spectrum is sensed by cryptochromes and also phototropins. Now we also have some other sensory photoreceptors which are involved in biological clock, and they are called Zeit loops, and there are 3 of them known in plant system. Interestingly, UVB, which is known to be very, very harmful otherwise, also regulates some aspects of plant development, and now we also know what the receptor for UVB light is. This is something interesting that we also have cryptochromes in our own biological clock, and although they were discovered for the first time in Arabidopsis, but to our surprise, all the Drosophila also have cryptochrome, which are sensory molecules for controlling biological clock. But human systems, although it's a part of the biological clock feedback loop mechanism, but they don't sense light to regulate the biological clock. This is something very, very different in animal systems or mammalian systems. And so in that sense, I think it must have evolved some new functions in our own biological system. Phototropins are unique to plants, and in fact I had some fundamental contribution to make here during my postdoc in Michigan State. I isolated the first ever mutant in phototropism, which later led to the discovery of this particular photoreceptor by Winslow Briggs at the Carnegie Institution in Stanford. Another thing I would like to mention also, somebody in the audience mentioned, or the speaker mentioned, about the work of Darwin in evolution. Some of you may not be familiar, and during the later part of his life, Darwin also worked on and also wrote a very famous book by the title, The Power of Movement in Plants. And he talked about phototropism, gravitropism, tendril coiling, things like that. And he was the first one to tell that the grass coleoptile, a seedling, bends towards light, and the light is perceived by the apical part, but the portion which bends actually is the subapical part of it, in a way that there has to be a transmission from the apical part to the lower part, which is going to be bending. And that turned out to be very interesting to find out that the first hormone auxin is actually involved in that. And so it has almost taken from 1880, when he mentioned about that, to 1997, when we had the first receptor which is the one which is responsible for sensing blue light to regulate phototropism, and that is what is phototropinase. Interesting part is that there are 5 phytochromes in Arabidopsis, 3 in monocots. There are 2 cryptochromes and 1 cryptochrome dash. There are also 2 phototropins. So there are small variations within these systems, and they may also perform different functions. Okay, what exactly do these photoreceptors do? I'll just go through this particular life cycle of a plant. In fact, they regulate practically every aspect of plant development, but some of these major features which are listed here are, right from germination, there is a role of red and infrared light, and this is sensed by phytochrome to induce seed germination. Once a seedling emerges, if you allow a seed germination to occur in dark, you will have an etiolated phenotype, but when you are exposed to light, this actually undergoes photomorphogenesis and leads to chloroplast development, and further on regular development takes place. And this transition, which is called de-etiolation, is controlled by red and infrared light through phytochromes, by blue UVA light through cryptochromes. Now, another aspect which is very, very noticeable in terms of the response of light is, if you actually have a very dense planted crop or in a field anywhere where you plant, naturally the germination is not going to be very, very uniform. Some of the seeds might lag behind. As a result of that, the ones which are actually taken up earlier, they might shade the younger ones. And this leads to what is called shade avoidance, and this shaded actually seedling tries to elongate much longer. And this is controlled by red and infrared light through a specific class of photoreceptors called phytochrome B and phytochrome E. And actually this is a wasteful process, and if you are able to actually knock out that particular gene, you are able to have a very uniform cropping pattern as such. Now coming to phototropin, which is responsible for causing phototropism to a unilateral light. This is controlled by a phototropin called phototropin 1. And another phototropin which is also involved in regulating another aspect in plant development is the chloroplast movement. Every cell of a leaf has about 100 chloroplasts, and they keep moving. And what really happens is, if you have no light at all, they are uniformly distributed all through the cytosol. When they're exposed to some limiting light, they actually orient in a fashion so that they can capture maximum light for photosynthesis. But if you have a very high energy of light, this could be actually very, very damaging through photooxidation. They tend to run away from that and causes protection from that kind of high intensity light, and this is a response which is called chloroplast avoidance response. This is now known to be controlled by another phototropin which is called phototropin 2. Now this lower response is controlled by phototropin 1, but the higher one is controlled by phototropin 2. Now another response which everybody knows, and I think all biologists know about, stomatal opening closing, which is very vital for plant development, oxygen exchange and other exchanges of the moisture also. This also is controlled by light, and within white light it is blue light which is very critical for that. It turned out to be that all these responses were known to be controlled by blue light for almost 70-80 years. But FORT1 mutation was only affected in phototropism. FORT2 mutation only caused a response which was lacking in chloroplast movement. But then these two mutations actually did not cause any change in somatal movement as such. At that point of time, we thought that probably there is going to be a third receptor. But it turned out to be that both are essential for that. If you have a double mutant, that also will be missing this particular response. So now we know FORT1 controls phototropism, FORT2 controls chloroplast movement, FORT1 and FORT2 together they control somatal movement. As I mentioned in the beginning also, this biological clock is entrained by light, and in case of plants specifically, we have a unique receptor called phytochrome, and like our own system, we also have cryptochromes. So both these photoreceptors sense light to entrain our biological clock, which is very critical also for transition to flowering, because that's the way these receptors sense photoperiod in the leaf and cause this transition to flowering. So both phytochrome, cryptochrome also are involved in transition to flowering, which is a very critical phase of life cycle, because without that we will not have any seed set, no harvest as such. So this is the overall background, giving you an idea that how critical these photoreceptors are in regulating plant development all through their life cycle. Now coming to some of these components which work through downstream to these receptors. In fact, there are dozens of them, but I only listed a few of them. So these are the receptors I just talked about. Downstream to them, there are these early signaling factors called PIF1, PKS1, PKS2. And downstream to that, I think this is one very critical factor called COP1, which is a component of E3 ligase involved in protein turnover. And downstream to that, another two important ones are Hi5 and H5H, which is a homolog of Hi5 in case of Arabidopsis. So this particular signal, I think, is turning out to be very, very important for all light signaling, whether it's in response to red light, far red light, blue light, or UVB light. Largely to the work done at Yale by Xinguang Dang, now we know that this COP1 interacts with another component called SPA1. Together, they recruit Hi5, and once they recruit that, it's able to cause ubiquitination, it will cause degradation. This all is happening when you have plants growing in dark. So because of this degradation of Hi5, there is no photomorphogenesis. So what is happening on exposure to light is, whether this is blue light or red light or far red light, SCOF1 actually is shunted out of the nucleus. As a result of that, you don't have any interaction of this SPA1 and COP1, and Hi5 is free. As a result of that, this transcription factor is able to go and bind to the motors of these genes and regulate gene expression, which leads to photomorphogenesis. You'll be surprised that from chip-chip analysis, Xinguang Dang has shown Hi5 binds to promoters of about 3,000 genes to regulate photomorphogenesis. Some of them may be downregulated, others may be upregulated. Now, the work in our lab, so this is the overall background about our knowledge about light signaling, which largely came to work under Arabidopsis. At that point of time, as Dr. N.K. Singh said, their group and our group at South Campus, Delhi University, we participated in the international program to sequence rice genome. And so this was published in Nature in 2005. The issue was of August 11th. Incidentally, in the same week, Science carried a news that this sequence of genome rice has been achieved. And in fact, they published this particular photograph where I happened to be there, although N.K. Singh was a partner, T.R. Sharma was a partner, my colleague was also a partner. Incidentally, this photograph was clicked when I was there attending a meeting in Japan, and they were not there. So this picture appeared in Science for that matter, but that is, I think they have equal contribution to sequencing of the rice genome as I had. Now, once this sequence became available, idea was to see whether how you actually utilize this particular information to identify genes and also their function. So as a 5-lab consortium, we undertook this particular project where we tried to understand genes which are regulating transition to flowering and panicle development as seed set. And 3 of our colleagues, Dr. Tyagi, Dr. Sanjay Kapoor, and myself, we took up this challenge along with Usha Vijayaraghavan in Bangalore. We tried to identify genes which are differentially expressing during various phases of transition. So we took 3 as control, which were vegetative, leaf, fruit, and seedling. We decided to choose the stages from foliar transition, shoot apical meristem, and 4 of other stages along with that. 7 stages we chose on during panicle development when the transition already taken place. We also chose to work on 5 stages of seed development right after fertilization, how the seed matures. And this all data which came through was very, very remarkable. It gave us an idea that how during different stages of development there is a differential expression gene occurring. That every stage there was a differential expression, but there were major spikes during development. And what are these stages which are representing overall floral development? This particular major jump occurs during organogenesis, followed by meiosis, polar mitosis, sperm cell differentiation, fertilization, embryo development, like that. So I think we came up with a very nice data which showed that there were major changes occurring in development of the panicle, and gene expression was getting highlighted during these stages. This particular heat map also shows that during panicle development stages, some of these transcription factors, their expression was changing during different from P1 to P6 stage. And looking at this list, it looks like some of the major classes are of DOF domain factors, PhD, SRF, BZIP, Polycom, and of course PL family, homodomain, and also SRF gene family. So I'm going to focus only on this particular transcription factor family, which my lab analyzed at the whole genome level. Also analyzed how differentially they are expressing during panicle development. This is one example here I'm showing. You can see some of these genes, BZ47, 62, 41, there are about 89 genes of BZ family in rice, and you can see that how the transition is taking place. They are not expressing much during vegetative tissues, but they are expressing during panicle development after the transition has taken place, and their expression goes down during seed development. And there are others which are not expressing at all in all these stages, but their expression is very, very high during seed maturation. This kind of expression, I think, was providing us a clue that they must be having a specific role to play during those stages of panicle and seed set in case of rice. Now, I'm not going to touch upon all these genes, but I'm only going to focus on now this Hi5, which I said is a basic transcription factor. And there's only one high 5 in case of Arabidopsis. So once the genome was available, we made this phylogenetic tree. It turned out to be that although Arabidopsis only has one high 5, it has one high 5 homolog. But in case of rice, we were able to identify 3 of them, BZIP1, BZIP48, BZIP18, which were homologous to ATH5. So the question was, which of them is actually more close to high 5 in function, or whether they have some kind of overlapping function, or they have acquired newer functions during the course of evolution in case of monocots like rice. So we chose to work on one of them, and which is BZIP48. And because this was easy to clone to begin with, that's why the work started in that direction much more than the other ones, BZIP1 and BZIP40 and BZIP18. Now, first thing to show is whether is it functionally equivalent to high 5 Arabidopsis or not. Easy thing was to be able to complement Arabidopsis mutant with this particular gene. So look at this particular phenotype. This is wild type Arabidopsis, which is about 4 days old, grown in light. And this is that high 5 mutant. When you knock it out, it causes elongation in the seedling height. And now you put this particular rice gene and make the complementation, you can rescue the phenotype, make it wild type. So in this respect, it's functionally equivalent to high 5 Arabidopsis. And if you express this particular gene in wild type, it does not make much of a change. At the same time, we also realized that there were some other traits also in this particular mutant of Arabidopsis. Look at this particular seedling position of the cotyledons. They are actually in this particular orientation here, and in case of the mutant high 5, they are vertically bent down. And this phenotype also could be rescued by the rice gene. And this is another trait which is also rescued. And look at the phenotype high 5 and the wild type. Look at this, the angle of the fruit, which is called silicua, like custard plant. This is a certain angle which is very, very steep along the axis of the stem in case of wild type, but this is much wider in case of the mutant high 5, and we could also restore that by putting the rice gene. Although this gene and the mutant has been worked out for almost 20 years, but to our surprise, nobody noted this particular phenotype, and we were the first one to report. This is also something which is affected in case of mutation in high 5 and could be restored by Arabidopsis gene. Now, as I said, if you knock out this particular gene in Arabidopsis, it causes elongation, and is it in a way also involved in controlling plant height? Everybody knows that what exactly the contribution of Korean evolution was, and Norman Borlaug got the Nobel Prize for that. This was also a gene which was involved in controlling plant height. These were the natural wheat genotypes at that point of time, and because of the mutation it causes some kind of semi-dwarfism, which led to a lot of stability in the plant and also higher yield, which led to green revolution. So we are also keen to know whether this light-induced gene also, or lighter gene also may have a role to play in controlling plant height. So next slide will give you some idea. This particular gene now, OsZIP48, we were able to overexpress in rice, and this is the wild type, and also these 3 independent lines of transgenics, which are overexpressing BZIP48, ortholog high 5. This causes drastic reduction in the plant height. But this is a surprising thing, because in case of high 5 in Arabidopsis, although when you knock it out, it causes elongation, but somehow when they overexpress that, it did not cause a reduction. And this is the change we are finding is happening in case of rice versus Arabidopsis. This is very, very dramatic reduction in the height as compared to other genes known in the literature which causes regulation of plant height. We also were able to see that, of course, this is now the internode of wild type. Within the same height, we have 2 internodes in case of the overexpression line, which is causing dwarfism. And there were also some changes also in the overall girth of the plant, and also changes in cell size and also cell length, which is also estimated here. So these kind of changes, I think we thought should have been very, very useful to give it some kind of a sturdy phenotype of the plant. It turned out to be the other way around, because this particular section shows that overall girth actually of the stem has reduced, and it's causing some kind of brittleness. Okay, brittleness to the stem may not be very, very useful overall for plant strength and overall flexibility. When you knock this particular gene out, you also find that there is elongation of the internode. Not only that, you also find aerial roots coming out of that, which is also expected, like in case of Arabidopsis. Now, this came as a surprise. We also got to obtain one TDN insertion line, and when we had the homozygous line, it turned out to be seedling lethal. In case of Arabidopsis, it is viable. When you knock the gene out, it actually becomes seedling lethal, which also is something, a unique observation we made in terms of this role in rice development. We also carried out microarray analysis of all these overexpression lines and also knockdown lines, and found out that there are different hormone pathways involved in GA biosynthesis,